In this video, we'll take a look at the structure and function of the gallbladder and bile. Keep in mind that the liver produces bile. Bile consists of bile pigments, bile salts, cholesterol, mucus, and water. Let's take a closer look at what bile pigments and bile salts are. Bile pigments are produced by the liver from the dead and damaged red blood cells that the Kupffer cells of the liver remove out of the circulation. Keep in mind that when the liver disposes of the red blood cells, that red blood cells don't have a nucleus and other typical organelles. Instead, these cells are filled with the molecule hemoglobin, which means that the liver is going to have to dispose of hemoglobin. And so each hemoglobin molecule contains four proteins. The protein can be metabolized by the liver into individual amino acids, and each of those four proteins contains a heme group. So taking a closer look at the heme group and how the liver can dispose of that heme group, remember that the heme group contains an atom of iron in the center. The liver is going to have to extract that atom of iron, bind it to a carrier, and save it for reuse. We need to recycle that iron to make more hemoglobin. The carbon frame that surrounds the iron atom, on the other hand, is a waste product. The liver is going to metabolize this carbon frame into bilirubin, a bile pigment. Bile salts, also known as bile acids, are part of the bile, and these structures are produced by the liver. Bile salts are amphipathic structures, which means that they're both hydrophilic and hydrophobic in nature. The bile salts are being depicted here as these green structures. So let's compare the bile salt to another amphipathic structure that we've already learned about, the phospholipid. Remember that the phospholipid had both a hydrophilic and hydrophobic portion. There are two fatty acid tails that are hydrophobic, and those fatty acid tails come off of a hydrophilic head group. So remember, the arrangement of phospholipids within the plasma membrane is that the fatty acid tails aggregate together within that watery solution, and so they create a fatty barrier in between two watery solutions, because inside of the cell, the cytoplasm is water-based, and outside of the cell, the extracellular fluid, that's also water-based, and that's okay because the hydrophilic head groups of that phospholipid bilayer make that fatty plasma membrane soluble in a watery environment. In the case of bile salts, if we put an amphipathic molecule into a watery solution like the inside of the small intestine, what will spontaneously happen is the hydrophobic region of those bile salts will aggregate together, and the hydrophilic regions will be soluble within the watery environment. And so here I'm drawing the bile salts like a phospholipid just to get this point across. So the way that these bile salts work is that the fatty acid tails, still, just like we saw in the plasma membrane, are going to aggregate together, and then the hydrophilic portions of the bile salts will be solubilized in the watery environment of the small intestine. This spontaneous organization is what's known as a micelle. So let's take a look at how bile salts are going to interact with fat in the small intestine, knowing that they will spontaneously form micelles. This type of physical breakdown of a big globule of fat into many small droplets of fat is analogous to chewing, right? It's just a mechanical breakdown. It's taking one large piece and dispersing it into many small pieces of the same thing. In other words, this is not chemical digestion. This is just a gross mechanical breakdown known as emulsification. Bile salts emulsify fats within the small intestine, which means that they take the big fat globule, break it into many small droplets, and this way, fat digesting enzymes, lipases in other words, are going to be able to work along a larger surface area. And so what emulsification is doing is it's increasing the surface area through which our fat digesting enzymes are going to be able to work. So now that we know what bile salts do, let's take a look at where bile salts go after they do their job. So in this picture, we can see that the bile salts are being released into the small intestine from the liver and the gallbladder. They're going to do their job of emulsifying the dietary fat that's within the small intestine, allowing that fat to be digested, and that fat is going to be absorbed into the lymphatic system without the bile salts. And we'll talk about that in a different video. But what I want to show you here is the bile salts, once they've done their job, here they are still in the small intestine and the fat has been absorbed separately. Those bile salts get absorbed back into the blood. And remember, that blood is the hepatic portal vein. That's a blood vessel that's going to take all the blood from the small intestine and return it to the liver. Once the blood from the hepatic portal vein enters the liver, it goes through the lobules of the liver, and there the hepatocytes will remove the bile salts and put them back in the bile. And so bile salts are recycled in that way. The liver is also able to make new bile salts from cholesterol. So to recap, we've looked at the composition of bile, what bile pigments are and how it's an excretory product. We looked at the nature of bile salts and how they emulsify fats. Cholesterol, mucus, and water act primarily as a buffer. Once bile is secreted into the small intestine, that bile, most of it, is destined to become eliminated from the body with the feces. Next, let's take a look at the flow of bile from the liver into a duct system and how the gallbladder connects to that duct system. Here I made the liver and the pancreas transparent so we could see the branching ducts inside of those organs. And we can also see how those ducts are continuous with each other, and they're continuous with the gallbladder, which I left solid here. Next, let's remove the liver and the pancreas from our view so we can focus better on the duct system. So here we can see the interconnected duct system without the liver and the pancreas. And so let's think through the production of bile, the flow of bile, and the role of the gallbladder. The liver continuously filters our blood, continually processes our blood, and so 24/7 the liver is producing bile. And that bile is collected in the liver by the right hepatic duct and the left hepatic duct. And those hepatic ducts merge to form the common hepatic duct that leaves the liver. Bile would then flow into the common bile duct, and the common bile duct connects with the pancreatic duct at this distal region called the hepatopancreatic ampulla. So the opening into the small intestine is regulated by a muscular sphincter called the hepatopancreatic sphincter. Typically, that sphincter is closed. And when that sphincter is closed, you can imagine what will happen is the bile that's in the hepatopancreatic ampulla can't get in there. And so the bile is going to start to back up. And it's going to back up into the common bile duct. And from there, instead of going back up into the liver, because there's a continual downflow of bile from the liver, it will take path B, the cystic duct. So when the hepatopancreatic sphincter is closed, bile is going to back up into the cystic duct, and the cystic duct is the duct that connects with the gallbladder. And so the purpose of the gallbladder is it just stores bile. It doesn't produce bile. It's a non-vital organ, as you know. Many people have the gallbladder removed. And so it just stores bile. Now, the hormone that opens the hepatopancreatic sphincter, so the hormone that would open the entrance of bile and the pancreatic secretions into the small intestine, is the same hormone that will cause contraction of the gallbladder. So automatically, when that hepatopancreatic sphincter opens, the gallbladder will contract, and the bile that has been being stored is ejected into the small intestine.